all of your cells have the same DNA, the same genes, but not all of your cells look alike. The reason is not all of your cells are using the same genes at the same time. Right? You have the ability to turn on certain genes and turn off other genes, express them when they're necessary and not when they're unnecessary, and that's how a liver cell looks different from a skin cell, looks different from a stomach cell. And even if you're a single-celled creature, you don't have to use all of your genes all the time, right? Sometimes you have a gene that makes an enzyme to break down a particular nutrient. Well, you don't need to make that enzyme unless that nutrient is present. And not only don't you need to, it's a waste of energy not to, right? It's sort of like, if you are Harry Potter, right? You have a spell book, right? You don't have to use all the spells all the time. You only use them when they're necessary. Otherwise, you're just wasting some energy. That might be a stupid analogy. But let's talk about single-celled creatures. Bacteria tend to have genes that need to be expressed at the same time, that need to be expressed together, next to each other. And they share a single promoter. So a promoter is that stretch of DNA that helps RNA polymerase bind to the DNA so that it can make an mRNA transcript of the DNA that follows. So they just have one promoter and then have several genes in a row right afterwards, so that when you make one gene, you make all of them because they are expressed together. Well, why would you need several genes expressed together? Well, for instance, if you have to make tryptophan, tryptophan is an amino acid that we can make ourselves as single-celled creatures, but it takes several chemical reactions, right? You start with a precursor up here, and then you have to turn that into the next intermediate, which you have to turn into the next intermediate, which you have to turn into tryptophan, right? So that is 3 different chemical reactions that have to occur. Each of those chemical reactions require an enzyme, right? And those enzymes are coded for by genes, right? So those genes, some of which have 2 polypeptides, right? So they need 2 genes. Those genes all need to be coded for at the same time. And so all 5 of these genes are on the same trip operon. So we have all of the genes, right? And then we have the promoter just before those genes, which helps RNA polymerase bind. And then right kind of either in the middle of the promoter or between the promoter and the genes, you have something called an operator. And that's essentially a switch that allows you to turn on and off these genes, right? You can either block transcription of this entire set of genes, or you can facilitate it. And we call that promoter, the operator, and the genes, all of those together are called an operon. That switch can be turned off by something called a repressor. And this repressor is just a protein that you code for elsewhere, right? It's a regulatory gene that you make. But generally that repressor has 2 different shapes it can be in, right? If it's in one particular shape, it can bind to the operator that blocks RNA polymerase from binding and turns off the gene. You can't make an mRNA transcript. Or it has another shape where it can't bind to the operator, and that leaves the switch on, right? And that allows you to make mRNA polymerase, right? Well, what causes it to go into the shape where it can bind to that operator and turn it off, right? Where it can repress, right? Well, that repressor needs to bind to something called a co-repressor in order to put it in the right shape to turn things off. So let's look at a picture of this. Here is our operon, right? We have our promoter and our operator and all of these genes. This is our operon. And normally, right, our repressor is inactive, right? It's a shape that can't bind to this operator, and so it can't block RNA polymerase. RNA polymerase can very easily bind to this, make an mRNA transcript that will code for all 5 of these genes. Because normally you have to make tryptophan. But if tryptophan's present in the environment, well, then you can just absorb tryptophan across your cell. You don't have to make it anymore. Well, tryptophan also, when it's present in the environment, binds to this repressor and changes the shape of the repressor so that all of a sudden now it can bind to this operator, and that blocks access of RNA polymerase to this set of genes, to this operon. So when tryptophan is present, it acts as a co-repressor because it activates this repressor, right? It binds to this repressor and puts it in the active shape, and then that repressor can then bind to the operator, this on-off switch, and prevent RNA polymerase from making that mRNA transcript. And if you can't make that mRNA transcript, you can't undergo translation either to make a protein, and your gene is not going to be expressed. It is turned off. Your TRP operon is one that is generally on, and it gets turned off in the presence of something. So it's called a repressible operon, right? It's normally on, but in the presence of tryptophan, it gets repressed. It gets turned off. Generally, where you're making something like an amino acid, you're going to want to make that as the default option because you need it to survive, and you'll stop making it when it's present in the environment. Generally, those kinds of things are going to be repressible operons. But you also have operons that are generally off, and you turn them on in the presence of something. These are called inducible operons. So if this is a nutrient that you need to metabolize, right, and what you make in this operon is a set of enzymes or something to break down that nutrient, right? Well, you don't want to make those by default. You only want to make those when that nutrient is present. So those are induced in the presence of that particular thing. And so one example of this is the lac operon. And the TRP and the lac operon are two kind of classic examples of this prokaryotic way of regulating gene expression. So in this case, instead of having a co-repressor, which binds to the repressor and makes it active so that you can block transcription, right? And you can turn that operator off, essentially. You have what's called an inducer, and the inducer binds to the repressor and inactivates it. And that allows gene expression to occur. So let's look at this. So here is our promoter and our operator, and then behind this would be your lac operon, right? You can see it right here. And I have several videos available for you to watch. Normally, you have an active repressor which binds to this operator and blocks RNA polymerase from binding to the DNA, and you will not make an mRNA transcript. The genes will not be expressed. They are turned off. That is your default setting. But if lactose is present, or in this case allolactose, which tends to be present when you have lactose, then it will bind to this repressor and inactivate it. And now, all of a sudden, when lactose is present, we can't bind the repressor to the operator. You can't block RNA polymerase, and so RNA polymerase can bind to this promoter, zip right past the operator, and make mRNA transcripts for these three genes, which are all necessary to bring lactose into your cell in large amounts and break it down. So this is an example of an inducible operon. Again, generally, enzymes that code for catabolic pathways or operons that code for several enzymes involved in a catabolic pathway that breaks something down, like lactose, for instance, are inducible operons, meaning their default stage is off, but if those nutrients are present, it induces you to turn on that operon. Things like the TRP operon, which produce, an amino acid or something like that, are going to be repressible operons, meaning you want the default state to be on. You are making this unless it happens to be present in the environment, and if it's present in the environment, that will repress production of that amino acid, right? So it will turn off that operator by activating the repressor. All right, we've talked about both repressible and inducible operons, but both of these rely on the use of repressors, right? Repressors which bind to the operator and turn the gene off. So they're both examples of negative gene regulation, right? Both inducible operons and repressible operons, right? The only difference between those two is that repressible are automatically on, right? Meaning the repressor is inactivated. Inducible are automatically off, meaning the repressor is normally active. And you have to turn it off in the presence of something. But again, you're using a repressor, so that's negative gene regulation. Positive gene regulation is using an activator instead of a repressor. And these two, negative and positive gene regulation can happen side by side. So for instance, with the lac operon, right, the lac operon uses negative gene regulation because it has a repressor, but it also can use positive gene regulation. So when lactose is not present, you don't want to make all those proteins that help you metabolize lactose. that said, when lactose is present, you may want to make all those things, but not necessarily, actually. Because when glucose is present as well, you actually prefer glucose to lactose. It's fine. You can metabolize lactose, but you prefer glucose. So when glucose is scarce, when there's not very much glucose, you tend to build up cyclic AMP, which is a molecule that we've talked about before. Well, cyclic AMP, if we look at here, binds to what's called an activator. Again, this is positive gene regulation. And that activator then takes on a different shape, and that's a shape that is able to bind to the promoter, and it doesn't inhibit like the repressor. It helps RNA polymerase bind to the promoter, right? So it causes you to make even more of these gene products. So when lactose is present, we've inactivated this repressor, so we can access this operon. And when glucose is scarce, we also have this activator that promotes transcription. And so we're going to make lots and lots and lots of these gene products within this operon. All right, well, let's say lactose is still present, right? So we can still access this operon, but glucose is present. Well, our activator is not active anymore because when glucose is present, we don't have nearly as much cyclic AMP, and so this isn't helping the RNA polymerase bind to the promoter. So you can still make some of these enzymes to metabolize lactose, but you're not going to make nearly as many. And so because we're using, in this case, an activator, right, this is positive gene regulation, as opposed to negative gene regulation, which is when we talk about using this repressor. So operons are a clever solution for having gene regulation in a way that allows you to express genes together very efficiently when those gene products are going to be used together at the same time. But that's really only found in large amounts in prokaryotes. So think bacteria, right? In eukaryotes, it's a little bit more complex, as you might expect. And there are actually lots of ways that you can regulate gene expression in eukaryotes. But first, you have to understand how genes are expressed. And we talked about this in the last chapter, right? Now that we know the whole process of gene expression, well, we can look at every spot along those pathways, right? You can interfere with the process and inhibit gene expression. So first of all, we know that DNA is often coiled up as chromatin, right? This chromatin, when it's tightly coiled, it's hard for RNA polymerase to access the DNA, and so those genes are essentially turned off. So this would be called heterochromatin. Now, if we unfold that, we make euchromatin. This DNA that's unfolded is now open to transcription, right? Then, so we'll talk about ways that we can regulate whether transcription occurs or not, aside from folding up the DNA. Then transcription happens, and we make an mRNA transcript, right? Well, we splice up in eukaryotes that mRNA transcript. That doesn't happen in prokaryotes. In eukaryotes, it does. Well, we can mess with that splicing process, right? We can cut out certain introns and not others, right? We can cut out introns and take those exons and put them back together in different orders, right? All of those things are going to lead to different proteins. We can also regulate this as it gets transported to the ribosomes for translation. We can break down RNA before it ever gets to the ribosomes. So, if you ever, maybe sent out an email that you regret, right? Well, here, your DNA made an mRNA of a gene that it thought it wanted to express, but now the situation has changed. We realize we don't want to express it. We can cut it up. Similarly, we can destroy proteins after we've made them, or we can fold them differently, or cut them up, or whatever. There are lots of ways to regulate eukaryotic gene expression, and we're going to talk about a lot of those in the next video.